So the data we're going to use today is by this 10x paper that when they first introduced Xenium as a technology. It's very nice because all these technologies are consecutive sections. So you can map the biology from single cell to spatial, either in Visium or in Xenium. And the two Xeniums are replicates here. So we can later see them spatially. So here there are consecutive sections, as I said. I think previously you have heard that Visium is at the spot level and Xenium is at the cell level, but it's not full transcriptome. So previously in Python community, we have spatial data now. We also have ST-align to register these different slices. We do have a missing of these read, access, and visualize of such data in Bioconductor. So now we fill the gap by having these three tools. So first, to introduce SpatialExperimentIO, I do acknowledge that there's molecular experiment, there are spatial feature experiment package for this kind of reading access. But here we want a minimal data that can basically just take the count matrix and the spatial coordinates. And later on, Dario will submit a pull request to include the polygons without the transcript level of details. Because as statisticians, usually what we care about are the units at the cell level. So currently there are 4 technologies, and I added one last week, is by Sickfish, by a pull request. So yeah, this will continuously grow as we have more and more spatial technologies. And then for STexampleData, previously we only had some mouse brain Visium data, but now we complete the chapter with Cosmic example, Merscope example, and StarMap Plus. And also for this paper that we're going to talk about today, there are 4 modalities. We include Chromium here as a single cell experiment in the STexampleData because it's just a perfect replicate of these 3 modalities. And then for ggspavis, the first version had plot Visium as a function. But now we added function like a plot spots to, it's called spots, but you can use it for either spots or at cellular level. We added some new QC plotting functions and also how to visualize the on-cluster text annotation in the reduced dimensions. There are 2 other functions that I'm currently working on. I have them independently from spatial experiment as an object. But my goal is to integrate them, and I didn't have time to finish before this. But I can show you how they look like independently, and it's up to the user to choose whether you prefer to have a more simplified input, or using spatial experiment as an input. So yeah, we also added another color bar that's more like from blue to red, so you see more contrast. And I call it Surat-like because I got the idea from Surat. So first, add the spot resolution for Visium. Here are some QC plots that you can use. I don't know if it's obvious enough, but there are some red dots on the first panel here. So for all the QC spots, you can see them, where are they located on the space. And also there are low abundance genes. And these QC criteria are just taken from the OSCA book. It's not accurate, so I really look forward to Dario's contribution on this Space Trooper. And then here, I need to talk more about this. So on the left is the vendor, so 10x run this data, and they used the full resolution TIFF to downsize it. So the full resolution TIFF you have is the same image as you would have for low res and high res. But on the right, when you have a fiducial framework like this, is usually run by downsizing the CETASys image. And this would cause a misalignment with the spatial coordinate and the image itself. So usually what I notice is that if the data is from 10x, then they would do it properly by downsizing the full res. But if it's from, for example, some experiment people set up, they would, by mistake, go to the CETASys, and then when you plot, you would have a misalignment. So yeah, here I want to say if you are working with generating the data, try to bear in mind that we do have to follow 10x guideline that you run downsizing to high res and low res from the full res to here. So here there are different categories that you can plot, either by continuous variable or discrete variable. For discrete, you would have a spatial cluster. For example, that's like the labeling of the clustering results. And then here it can be gene expression or it can be metadata in the call data. And I also have another one that's not shown here is the scatter pie, which is to overlay the pie chart with the cell type proportion on the H&E image. Yeah, so here we start by plotting these. So with this function, I added some functionality of zoom. So you can zoom into the region of where the spots will be located at. And you can say that you don't show the coordinates. So now it's just the image. And then you can overlay some empty spots on it. So this function is currently in progress. How to integrate it with spatial experiment. And here, now you can plot the counts, UMI counts, which is library size. Yeah. On the top, I think at the previous step when we QC, we removed them because of low counts. Yeah. Yeah, no problem. And here, this is by this gene ERBB2. So you can see the distribution of it, but always one function. And then categorical, so this is a Bayes-based clustering, which is a Bioconductor package. I run it, I got the result, and then here you can see that the blue outline, the cluster 4 and 12, they are more, later you will see is tumor-based, and then the gray outline is DCIS. I got that information from here. So I run deconvolution and I project the cell type proportion onto this scatter pie. You probably have seen this plot without the H&E image, but I think it makes more sense now if you put them on top of each other so you can see the correlation between morphology and the cell type. Yeah, so now we move on to Xenium, and I'm still using the same replicates. So you will see how similar the structure look like. So here on the left is a replicate 1, on the right is replicate 2. And you can see they are not perfectly aligned. Probably when they generate the data, there are some shifting. But you can see you do have some structures that are similar between these two replicates. And you can get these plots by running this function. And then you can try to do some labeling transfer. So here I run single R and I migrate the single cell labels onto the space. And you can see that here, this region and here are like Visium before that are DCIS or invasive tumor. And now we can also visualize this on UMAP. And we can annotate these clusters. Yeah, so that's everything for now. And the next step is we're trying to integrate what we have here into this best practices book. And there will be 2 new chapters on cancer with Visium and Xenium. And I hope everyone can contribute because this is a book that we're trying to push it out within the next 1 year or 2. We're still missing quite some chapters on deconvolution, on clustering, and also some spatially relevant techniques such as like QC or some philosophical chapters that everyone can contribute. So yeah, that's the end of my presentation. And now you can go to GitHub to my repository that I created for today. So my GitHub ID is EstellaD, ESTELLA. And then the first repository, EuroBioC. Maybe I can go there. I can show you where it is. So if I hope it's big enough, you can see it. Yeah, so STELA-D and then spatial transcriptomics workflow. Yeah, so if you have time, you can try to clone it. Otherwise, I will just flash it through because I still have 16 minutes and I do have a lot of content. So if you clone it down, you will see a document like this. So here are the required packages that you need to install, and then you load the packages. So to start with, I will talk about Visium.io. Actually, is the workshop that Marcel will give tomorrow. So we migrated the reading process of Visium into this new package, Visium.io. And here are the functions on how to read it, but we're currently changing quite a bit the gene names and also the format that you can take. And then afterwards, you can convert this object, 10x Visium, into a spatial experiment. And afterwards, I will talk about SpatialExperimentIO. So here, for taking Xenium as an example, this is the output structure that you need. And I stored all the mock download in this pathway in the current repository. So I manually created a H5 file that is only 4 genes and 6 cells. So that is really small that you can easily practice the reading with. There are many other technologies such as Cosmix, StarMap Plus, and VisGen. So all of this you can read more in this package, SpatialExperimentIO. And then the next step for storing the data. So today we're mainly dealing with these 4. So you can call their name by Jane Sick is the author of the paper, and then breast cancer by Chromium, Visium, and Xenium replicate 102. So next, you can have a look at these files. For example, here you see the SCE, the single cell experiment class, and we have the annotation. So the original author computed some cell type annotation for this single cell data, and I just mapped it into this object, and I stored it in the ST example data. So you already have the annotation here. And I wanted to see the frequency, so I made a bar plot. And this is how it looks like. So you have most of them as an invasive tumor. And then Python macrophage 1. Yeah, this is ranked by the frequency of cell type. Yeah, we also have our Visium and Xenium objects. So yeah, this empty spot, I'm still working on it to make it spatial experiment compatible. Then you can add the library size into the metadata, and then you can visualize them spatially. You can also visualize them on top of H&E. And this is without H&E for the gene expression. This is with. And I add another functionality of showing the axis, because sometimes you might want to crop into one region, but you don't know what the X and Y limits should be. So now you can see like where are they. X here is blocked. Yeah, and this is the Bayes-Space clustering result. I stored it as a CSV, but if you run the tutorial, you would actually store it already in the metadata automatically. So here I wanted to, because the Bayes-Space was run on QC object, and this Visium object, we take it from the STexampleData, which is raw data. So I did a bit of QC just to remove the spots that weren't used for clustering. And then you can see that spatially is like what I had in the presentation before. And then this is a deconvolution result. And I haven't wrapped them into this spatial experiment yet, but I have it here. So this is the idea that I have, that you just take the scaling factor, you take the image without forcing them into an object. And from here, you would compute the high resolution coordinates by downsizing the scaling factor. And then you would plot the layer of the image, like the previously I showed you. And then you would load the deconvolution fraction and specify the radius. And here the function we're using is called geom_scatter_pi. So for this one, you can, after running plus the previous layer of just image with the scatter_pi, then you would get what I had in the presentation before here. Yeah, so yeah, I think I will make it flexible that the user can choose what kind of formats they want. I think this is what I just showed you is more straightforward, that you don't need to think about these objects, but I'll also make it possible for just integrating with the class. Then we move on to Xenium. So for Xenium, we are reading the data, and we can have the summary of these two replicates, what are the library size. Because they are not completely the same, so there are some differences in the library size distribution. And you can also plot the spatial library size of these two. It might take a while to run. So yeah, these two. And then we pre-computed a single R result. I store them as a CSV here. Go up. Yes. So there are two replicates. The left is a replicate 1, and then the right is a replicate 2. Yeah. Yeah, so you see like the structures are similar, but there were some shifting. Yeah. Why is it like this? This, I think, is some error by the not biologists, sorry. It's by the people who generate the data. So yeah. Yeah. So there are some tools to do that, to register. STAlign, for example, that's a Python tool. SpatialData, I think, automatically handles that. Yeah. Yes. Yeah. So this I'm not sure, because the idea behind this is very simple. You just manually select 3 dots between the 2 images, and you like the landmarks, and you align the landmarks. So in R, for example, in our lab, we have a Shiny app to do so, but it's only linear transformation. But if you want to deal with the twisting or the rotation of the image, then I think STAlign has an additional step of correction with nonlinear transformation. So yeah, I think this field is a mix. You can't really isolate to bioconductor only. Yeah. Yeah. Yes. Yeah. So I call it plot spots, but maybe the name is open to discuss. I think. Just a question. Yeah. No, it's always plot spots. Yeah, because I think the cells can be understood as spots as well, and I want to simplify, the naming. And then after I loading the label transferred from single R, looks like this. So each cell name would have one cell type label. And I subset it to the post QC object. And then I plot them spatially, as you have seen before. I chose this palette because the stroma are more just like background color, and the tumor is more standing out. And here I tried both. I think in my presentation before, it was IC transform. So you can see the isolation is not so well. But now if you check the UMAP I would have here. Let's see. Is more isolated. I don't know if it's meaningful biologically, like log normalization is better, but I don't want to say that. That's my PhD, so yeah, I'll need to investigate more. So yeah, but you do see that there are differences in the UMAP with different pre-processing steps. So that's my presentation. If you have any questions. Yeah. Yeah.
Something here. Are there any more questions?
Plot polygons. I asked Dario yesterday, so he has that in Space Trooper. I think I think the Samuel from the poster, he mentioned that there is a package called Voyager that already handles that. So I would like to try that and maybe incorporate it in here. Yeah. So like single R is more for single cell, right? There should be some other method that's like spatial relevant. I don't know which one yet. Yeah, but I think definitely we want something that's yeah, for sure. So for the workflow, it will just be a demonstration of what you can do. But yeah, like you can see, I just load it as CSV. So whatever cell type annotation you want, it can be incorporated here. Yeah. Yes. Which one? Spatial experiment. It's in the submission process. Yeah. So I think my contribution ID is 3370. Yeah. So you can check that. Yeah.
Yeah, I can have a question.
Yeah.
You mentioned a couple of times that certain things are still up in discussion.
Yeah.
Somebody wants to get in touch with you. What is the best way to contact you via the GitHub?
Yeah, I think with the issue or pull request or just I don't know if GitHub has a function of messaging. Yeah. Yeah, or email.
On the general channel of the site for the conference.
Yeah. Mm-hmm. Yes.
So I think this project of writing is very valuable.
Mm.
I think potentially it could be like Oscar.
Yeah.
So in the technical advisory board of Bioconductor, we have these things that sometimes work and sometimes don't work. It's working groups. So I was wondering if this is right solving working trying to push out the community. So maybe
Started working.
Start the work.
Yeah, sounds good.
Because I think one of the problems with such fancy projects is quite a bandwidth.
Yeah.
That you said like now you're waiting for that part like yours. It's super that it's feature experiment, Voyager. So there's a lot of different players that I think the impact of the book would be much better. I think we all sort of like sit together and try to be as inclusive as possible.
Yeah, definitely. I think now there are so many different packages to read. For example, there should be one chapter of this book just to suggest, like if you only want to load single cell level, go with SpatialExperimentIO, or if you want more molecular based, then go with ME. Yeah, so it's not saying that one should incorporate the other, but they just should stand alone, and then we point to different groups based on their interest. Yeah.